Hello, everyone, and a huge, warm welcome to this explainer. I am absolutely thrilled to be your guide today on what is going to be a magnificent, microscopic journey into the world of genetic testing. We're going to explore a technique that honestly sounds straight out of science fiction, but it's actually a vital tool used in labs every single day. If you've ever wondered how we can peer into the very building blocks of life, well, you are in the exact right place.
It all begins with a pretty fundamental question. How do scientists literally light up our DNA to spot genetic clues? I mean, think about it for a second. DNA is infinitesimally small. It's packed tightly into our cells, completely invisible to the naked eye, and incredibly complex. We can't just open up a cell and read a book of genetic code. We need a way to illuminate the specific chapters, pages, or even individual sentences of our genetic instruction manual to see if everything is exactly where it should be. We literally need to make the invisible glow.
To pull that off, we turn to a remarkable technique with a rather long, intimidating name: fluorescence in situ hybridization, or as it's universally known by scientists, FISH. Now let's break down what that actually means, because it makes perfect sense once you do. Fluorescence refers to the glowing markers we use. In situ is just Latin for in its original place, meaning we're looking at the DNA right where it naturally lives inside the cell. And hybridization? That simply means binding a specific probe to a matching DNA sequence. Put it all together, and FISH is an elegant technique using fluorescent probes to vividly visualize targeted DNA sequences right inside the cells.
Okay, let's dive right into this. Section 1, Decoding DNA with FISH.
When we prepare a cell for FISH, we aren't just creating a random wash of colors. We are creating highly specific visual markers. First, we use a specialized dye called DAPI. You can think of DAPI as our background canvas. It stains all the DNA in the cell a brilliant, glowing blue, revealing the overall landscape. Then, because we can use multiple FISH probes at the same time with different fluorescent markers, we can track several sequences at once. For instance, we might use a glowing green probe engineered to bind exclusively to a sequence on chromosome 9, working right alongside a radiant red probe bound specifically to chromosome 22.
Moving right along to Section 2, Metaphase. Condensed and clear.
During metaphase, the cell is gearing up to divide. To do this efficiently, the DNA coils up tightly and condenses into distinct chromosomes. I actually love to think of this like packing for a major trip. You wouldn't just drag all your clothes loosely through the airport, right? No, you'd neatly fold and pack them into tight, distinct suitcases. That is exactly what the cell is doing here. The genetic material is packed into these dense, distinct suitcases that we call sister chromatids, which makes them incredibly clear and defined under a microscope.
This phase brilliantly illustrates just how dynamic cellular biology really is. During metaphase, we're looking at DNA that has already undergone replication, meaning those sister chromatids are present. But their visual appearance isn't always identical. Depending on the precise moment the cell is frozen in time for our test, they can look slightly different. Sometimes they're a bit less condensed, showing two distinct separated arms. Other times, they're bound so tightly together that they might appear as a single, unusually thick chromosome arm.
So the absolutely crucial point here is how our fluorescent probes interact with this replicated DNA. Because the DNA is already duplicated to form those sister chromatids, our probes are going to light up twice. The signal is highly localized to a specific region on the chromosome, giving you two brilliant signals per chromosome, one glowing beacon from each sister chromatid. For geneticists, this provides incredibly precise, double-verified visual data about exactly where that specific genetic sequence is located.
Let's shift gears to Section 3, Interphase, the diffuse nucleus.
If metaphase is a set of neatly packed suitcases, interphase is an unpacked, totally messy drawer. In this phase, the DNA is decondensed. It is unfurled and spread out everywhere to allow the cell to actually read the genetic instructions it needs to function day to day. When we apply our DAPI stain to an interphase cell, we don't see those distinct suitcase structures anymore. Instead, the decondensed DNA fills the entire nucleus, creating a vast, diffuse, glowing blue signal that takes up the whole space.
Now, what's really interesting is the direct contrast between these two phases. In metaphase, we have those crisp, localized double signals on structurally distinct chromosomes, but in interphase, the DNA hasn't replicated yet, so when our red or green probes find their target sequence, each chromosome is represented by just a single, isolated spot. It looks kind of like a lone, glowing star against the cloudy, diffuse blue background of the decondensed DNA. Which naturally brings up a highly practical question.
If these two phases give us such drastically different pictures of the exact same DNA, why do doctors choose one method over the other? We've talked about the pure biology, but how does this abstract, glowing science actually translate into real-world medical applications for patients?
Let's find out in Section 4, Rapid Answers, Real Diagnosis.
It all comes down to some crucial trade-offs. Metaphase fish undoubtedly offers far more detailed visual information. You can see the actual chromosome structure and pinpoint exactly where a signal is located. However, it's demanding. It requires coaxing the cells into a very specific, actively dividing phase of the cell cycle, and that takes precious time. Interface fish, on the other hand, offers less specific structural detail because the DNA is so diffuse. But its superpower? It doesn't require the cells to be actively dividing. You can test a huge variety of cells right away.
And that leads us to the major advantage of fish as a whole. Both metaphase and interface fish are primarily utilized as rapid tests. Because the probes bind directly to the DNA sequences and immediately light up under the microscope, these tests can be completed in a very short time frame. When a patient is anxiously waiting for a critical genetic answer, perhaps concerning a scary cancer diagnosis or an urgent prenatal screening, getting preliminary data quickly is absolutely vital.
However, we always have to ground this incredible science in medical rigor. While fish is amazingly fast and highly targeted, the results it provides are technically considered preliminary. Catching a glimpse of a glowing probe isn't quite enough to close the book. The rapid insights provided by fish must subsequently be confirmed, usually through a more comprehensive karyotype test or a highly detailed microarray test. It's those comprehensive follow-ups that ultimately provide the patient with their definitive official diagnosis.
So fish gives us a fast, brilliantly illuminated window right into our cells, allowing us to ask specific questions and get rapid answers by literally lighting up the dark space of the nucleus. But it's really just the first step in a much larger investigative journey. So I'll leave you with this thought. If fish gives us rapid answers by lighting up just a few targeted genes, what other vast, uncharted secrets are waiting to be confirmed in the billions of letters that make up our genetic code? Thank you so much for joining me on this explainer. Keep asking those big questions, and I'll see you next time.